Every living organism on Earth contains genetic material of some sort. In humans, this genetic material is called DNA, and it is stored within 22 non-homologous chromosome pairs and 2 sex chromosomes. However, chromosomal abnormalities can occur and cause problems. One such abnormality is chromosome translocations, and these involve the movement of genetic material from one non-homologous chromosome to another. The resultant translocated chromosome can lead to an increased risk of cancer and fertility complications. The reciprocal translocation is one of the two main types of chromosome translocation. This involves a section of one chromosome breaking off from the rest of the chromosome and binding to a second chromosome, which has also lost a section of genetic material. The first chromosome will then bind to the section lost by the second chromosome, completing the reciprocal translocation. A reciprocal translocation occurs after a double-strand break arises in the DNA. If this break is not repaired fast enough, the true chromosomal sections can drift apart, leading to the translocation. When the sections of the chromosomes finally come into contact, they bind when the double-strand break is repaired via a mechanism known as non-homologous end joining. The double-strand break which leads to reciprocal translocation can be caused by a multitude of factors, both endogenous and exogenous. Usually this break is repaired quickly. It is only when the repair is unable to be completed or even commenced that translocation is possible. The repair mechanism relies on two main proteins, Ku70/80, which binds to the end of the double-strand break and acts as a scaffold for the other proteins to bind, such as the DNA ligase protein, which is responsible for binding the incoming DNA bases together and reform the DNA backbone. Here are two chromosomes, and if we zoom inside this pink one, we can see the DNA, with the damaged DNA bases in purple. This DNA then breaks, leading to the double-strand break. The chromosomes then split into two sections, and each section ends up closer to the opposite chromosome. Then if we look back at the DNA, the Ku70/80 protein arrives to start the non-homologous recombination. This then signals for the DNA ligase protein to arrive, as well as nucleotides required to rebuild the DNA strand. The DNA ligase then seals the nucleotides together and the DNA strand is fixed, however, with the incorrect chromosome section leading to the two translocated chromosomes, as we can see here. The other main type of chromosomal translocation is the Robertsonian translocation. Humans have three different types of chromosome, all depending on the length of the P-arm, which is the short arm of the chromosome. Metacentric chromosomes have P-arms and Q-arms of the same length, with the arm separated by the centromere. Submetacentric chromosomes have a shorter P-arm than Q-arm, and acrocentric chromosomes have an extremely short P-arm, resulting in the centromere being very close to the end of the chromosome. Robertsonian translocations only occur between the acrocentric chromosomes. In humans, chromosomes 13, 14, 15, 21 and 22 are acrocentric, and here we can see the difference between submetacentric chromosomes and acrocentric chromosomes. 1 out of every 1,000 babies born is affected in some way by Robertsonian translocation, making it the most common form of chromosomal translocation. During the Robertsonian translocation, the centromeres of two acrocentric chromosomes fuse so that one large chromosome containing the genetic material of both chromosomes forms. The tiny P-arms of both acrocentric chromosomes are usually lost during the translocation process, but as they contain such little genetic material, this has no effect on the individual. Here we see two acrocentric chromosomes. The centromeres of both chromosomes make contact and both discard their respective P-arms, allowing the centromeres to fuse and one large chromosome to form. While translocations rarely impact an individual, problems often arise when a translocation carrier tries to have children. Normally, the child of two parents should contain 2 copies of each of the 22 autosomes, plus the X and Y chromosomes, and therefore 2 copies of each gene. However, when one of the parents carries a translocation, there is a chance this translocated chromosome is passed on to their child. As a result, the gene copy number will be incorrect, which can cause difficulties with the child's development and can often be fatal. Usually, a child is born with 2 copies of each chromosome, one from the mother and one from the father, as we can see here, with 1 green and 1 black chromosome coming from both the mother and the father. However, when a translocation is present, the translocated chromosome may be passed on to the child. as shown here, leading to 3 copies of the black chromosome. There are many disorders caused by chromosome translocations. Trisomy 21, also as Down syndrome, is caused by 3 copies of chromosome 21, and is often caused by Robertsonian translocation between chromosome 14 and 21. Trisomy 13, known as Bristol syndrome, is caused by 3 copies of chromosome 13, and similar to trisomy 21, it is also caused by Robertsonian translocation between chromosome 13 and 14. Individuals with Down syndrome have some birth defects and development issues, with varying severity, whereas babies suffering from Patel syndrome will not survive long after birth. Translocations often cause an increased risk of cancer. A common example of this is the Philadelphia chromosome, caused by reciprocal translocation between chromosome 9 and chromosome 22. This leads to an overexpression of the ABL gene, which causes unregulated cell growth, and it is a direct cause of chronic myeloid leukemia.